It's becoming more and more common to see things like gluten-free pizza or gluten-free buns at restaurants, grocery stores, and other food-based businesses. And this is partly because there's this increasing recognition, awareness, and diagnosis of a disease called celiac disease, sometimes called celiac sprue. As many as 1 in 100 North Americans are diagnosed with this. Celiac disease is currently understood as an immune system mediated disorder where the gluten in food triggers the body's immune cells to attack the cells in the small intestine. Gluten's found in all sorts of wheats and grains, including rye and barley. If we take a look at wheat, you've got your individual wheat kernels, and then inside each kernel there's this endosperm, which has a bunch of nutrients for the seed's embryo, mostly protein and starch, and some vitamins. The type of protein here is gluten, which is the main culprit in celiac disease. Well, really the main culprit behind celiac disease is a 33 amino acid peptide component of gluten called gliadin. Okay, got it. So the gliadin in gluten is what triggers immune attack in celiac disease, hence the need for gluten-free pizza or buns in hamburgers, right? Well, that's not the whole story, since we've known about celiac disease for a while now, like 70 years. So why the recent push? Well, within the last few years, there's been proposals of non-celiac gluten-related disorders, like gluten sensitivity, where gluten is thought to cause GI symptoms that seemingly improve when gluten's taken out of the equation. Unfortunately, as of this moment in February 2016, there's very little solid scientific evidence to back a removal of gluten from the diet unless you have celiac disease, although some people do seem to have improved GI symptoms on a gluten-free diet. Whether that's from removing gluten specifically or more broadly in adherence to a diet that's typically much lower in processed foods and carbohydrates remains to be seen. Alright, let's get back to celiac disease though. So, if somebody with celiac disease eats a wheat-based pizza, it's broken down in the stomach into gliadin and a whole lot of other stuff. Gliadin's a tough little bugger though, and resists being broken down by all sorts of enzymes. When gliadin gets to the small intestine, it's bound to secretory IgA in the mucosal membrane, which is an antibody that helps protect gut epithelial cells called enterocytes from toxins and pathogens. Usually, things bound to secretory IgA are marked for immune cell destruction, but in celiac disease, this gliadin IgA complex, for some reason, binds to a transferrin receptor, TFR, which is usually used to help absorb iron and seems to be overexpressed in patients with celiac disease. Once bound to this receptor, it's transcytosed across the cell from the apical to the basolateral membrane, or across the enterocyte and into the lamina propria, which is the thin layer that lines the gut wall. Once there, an enzyme called tissue transglutaminase, or TTG, cuts off an amide group from the protein. Deamidated gliadin's then eaten up by macrophages and served up on its MHC class II molecules. Remember, macrophages are in the gut and are always doing a bit of gut sampling, where they grab proteins, which a lot of times are from the food we've eaten, and show them to the immune cells. MHC stands for Major Histocompatibility Complex, and is the name of the serving platter for the stuff that's served up. And this is a normal way to make sure that there are no pathogenic bacteria lurking in the gut. Now, there are a ton of different types of MHC class II serving platters, and these serving platters are encoded by genes called human leukocyte antigen genes, or HLA genes. And these genes determine what things the MHC class II molecules serve up. So, for example, HLA-DR encodes for an MHC that serves up something different than the one HLA-DQ encodes for. Researchers have noticed that patients with celiac disease typically have specific deamidated gliadin serving platters, such as one called HLA-DQ2 or HLA-DQ8, which is an interesting clue that kind of helps us better understand celiac disease. It's at this point, when the HLA protein serves up gliadin, that the immune system kicks in. The macrophage throws it up top and is like, hey, guys, what do you think about this molecule? And the T helper cells, also known as CD4 plus T cells, from the immune system that recognize the gliadin zoom over and are like, yep, I'll take it from here. And they release inflammatory cytokines, which are molecules that initiate inflammation, like interferon gamma and tumor necrosis factor, which can directly damage and destroy epithelial cells in the villi of the small intestine. Not only that, though, the helper T cell stimulates B cells to start pumping out IgA antibodies against the gliadin, the transglutaminase enzyme, and endomysial antibodies, or EMAs, which are actually another type of antibody for transglutaminase, but for transglutaminase found in the endomysium, which is this layer of connective tissue around muscle fibers. It's not totally understood why these are produced, since they don't seem to cause any muscle-related damage or symptoms. It's possible they're produced just because of their structural similarity to the transglutaminase in the lamina propria. One thing they are helpful for, though, are making a diagnosis. Finally, the helper T cells also recruit killer CD8 plus T cells, which is when things start to get nasty. Killer T cells are drawn to and destroy cells undergoing inflammation. So, in short, as patients eat gluten, the immune system is stimulated and epithelial cells are destroyed. It's also possible that the destruction of these cells lets more glide in across the epithelium, since they're not bunched together as tightly as they were before. Tests for antibodies in the blood can be used clinically to help screen for celiac disease. IgA blood tests for both TTG and endomysial TTG can be effective ways to determine whether someone has celiac disease, especially in more severe cases. Although for more common, mild forms of celiac, these tests are less effective. Tests for IgA or even IgG antibodies against gliadin may also be used. For reasons that aren't well understood, a small amount of patients are IgA deficient, meaning they aren't able to produce any IgA antibodies in general, so in this case they'd need to have an IgG screening test done. Now this whole fiasco mainly happens in the duodenum, probably because it's the first part of the small intestine, and so it's the first to be exposed to gluten. The jejunum and ileum are also involved, but the duodenum is where most of the damage goes down. So a biopsy of the duodenum shows pretty clearly the extent of the damage. Healthy duodenum should look like this, where you have these really tall villi and these crypts that don't go down too far. With celiac disease, these villi can be destroyed and flattened out, called villus atrophy, and the crypts can get longer. These changes are called crypt hyperplasia, possibly from infiltration of immune cells or from the remodeling process that begins to take place during chronic inflammation. You'll also be able to see this infiltration of immune cells, or lymphocytes, in the epithelium. An endoscopic biopsy of this tissue can be an effective, yet more invasive way to diagnose celiac disease. So children with celiac disease often present with symptoms like abdominal distension or bloating, as well as this failure to thrive and diarrhea. Adults typically have chronic diarrhea and bloating as well, but the symptoms vary wildly. Also, patients can frequently have dermatitis herpetiformis as a complication, which actually has nothing to do with the herpes virus. What it is a bumpy skin rash that pops up from circulating IgA antibodies in the blood, where they mistakenly bind to the transglutaminase in the dermal papillae of the epidermis. Once they've bound, neutrophils swing by and start up an inflammatory reaction that's noticed on the skin as this rash. Okay, now, the main bad guy in this whole story that provokes the immune system and causes this whole mess is gluten. So symptoms and issues typically resolve when patients adapt a gluten-free diet, right? For some patients that start to adhere to a gluten-free diet, even for many years, there's still an increased risk of refractory disease, like small bowel cancer and T-cell lymphoma. Presumably, this is due to inflammation and immune system activation over time. Thanks for watching. You can help support us by donating on Patreon, or subscribing to our channel, or telling your friends about us on social media.